Hello, it's Dmitriy again, and I'm here to do Chapter 36 of Guyton Hull's Physiology textbook, the Pocket Physiology textbook in particular, for Chapter 36, Blood Types, Transfusion, Tissue, and Organ Transplantation. So we'll get right into it. O, A, B blood types. The antigens type A and type B occur on the surfaces of red blood cells, RBCs, in a large proportion of the population. These antigens, or agglutinogens, cause blood transfusion reactions. It is on the basis of the presence or absence of the agglutinogens on the RBCs that blood is grouped for the purpose of transfusion. When neither A nor B agglutinogen is present, the blood group is type O. When only the type A agglutinogen is present, the blood group is type A, and when only the type B agglutinogen is present, the blood group is type B. When both A and B agglutinogens are present, the blood group is type AB. When type A agglutinogen is not present on a person's RBCs, antibodies known as anti-A agglutinins develop in the blood. When type B agglutinogen is not present on the RBCs, antibodies known as anti-B agglutinins develop in the plasma. Type O blood contains both anti-A and anti-B agglutinins, and type A blood contains type A agglutinogens and anti-B agglutinins. Type B blood contains type B agglutinogens and anti-A agglutinins. Type AB blood contains both type A and type B agglutinogens, but no agglutinins. If that makes any sense, right? Basically, if you're type A, you're going to have a hostile reaction to type B blood because you're going to react to those antigens on the type B blood. But if you have type B blood, you're going to have a negative reaction to type A blood, i.e. blood that has type A antigens on it. If you have type O blood, which means you have no native antigens, you're going to have a negative reaction to both the type A and the type B antigens on type A or type B or type AB blood, essentially. But if you receive type O blood, which has none of the above-mentioned agglutinogens or the antigens, then if you're type A or you're type B or you're type O, you can receive type O blood because it doesn't contain any of the antigens mentioned. Therefore, will have no reaction to any antigen present because it doesn't exist there. The agglutinins are gamma globulins of the immunoglobulin M and immunoglobulin G immunoglobulin subclasses. The origin of the agglutinins in persons who do not have the antigenic substance in their blood seems to be entry into the blood of small numbers of group A and group B antigens in food and through contact with bacteria. When blood samples are mismatched so that anti-A or anti-B plasma agglutinins are mixed with RBCs containing A or B agglutinogens, the RBCs agglutinate into clumps. These clumps can plug small blood vessels throughout the circulatory system. In some cases, the antibodies induce lysis of RBCs through activation of the complement system. One of the most lethal effects of transfusion reactions is renal failure. The excess hemoglobin from the hemolyzed RBCs leaks the glomerular membranes into the renal tubules. Reabsorption of water through the tubules causes the hemoglobin concentration to rise, resulting in hemoglobin precipitation and subsequent blockade of the tubules. So as you can see from this table right here, table 36.1, blood types with their genotypes and constituent agglutinogens and agglutinins. Again, the agglutinogen is the actual antigen on the blood cells, and the agglutin is sort of this reactive entity against the blood cells. So again, if you have, let's say, genotype is OO, right? Therefore, the blood type or the phenotype is going to be O, your type O blood. Then you're going to have agglutinogens absent, right? Because there's no agglutinogens on, there's no antigens present on the blood, but you will have agglutinins or reactive entities against all the antigens that it could be possible, A or B, right? If you're OA or AA genotype, then because it is A is is dominant, it is heterozygous dominant, you will be still type A blood. So if you're AA, then it's obvious you're homozygous, but if you're OA, well, the A dominates, and so you will have A antigens present, agglutinogens present, and as a consequence, because you don't have any B antigens native, you will have anti-B agglutinins. And vice versa, if you're OB or BB, you will have B phenotype, your type B blood, therefore you have B agglutinogens, but because you don't have a native A agglutinogen, you will have anti-A agglutinins, right? Because it's a foreign entity to the body. It will react negatively towards that being present in the body. It'll try to lyse the cells, it might cause clumping, that kind of thing. If you're AB genotype, that means because they'll be co-dominant, and so you'll have AB blood, that'll be your phenotype, and so you'll have agglutinogens, i.e. antigens on your blood, on your RBCs of both A and B, but you will have no agglutinins because everything is native to the body of those kinds of antigens, and so you will have no negative reaction to either of them. And that's why, for example, that a person who is AB, type AB, or an AB phenotype can receive blood from any person, AB blood, B blood, A blood, or O blood, because they have no agglutinins or negative entities reacting against other people's blood because they just have all the antigens present natively. Vice versa, I will go further, in AB blood, a person who has AB blood really can't donate to anybody except other people who have AB blood because they have all the antigens that you could possibly have for the blood. And as a consequence of that, unless you have all those antigens as well natively, i.e. you also have AB blood, then you can't donate to that person. Again, vice versa, OO, you have type O blood, you can donate to any person because you don't have native agglutinogens, you have no native antigens present, so their body's not going to react to anything because it's not there, but you will have a negative reaction to anybody else's blood because you are reactive both against A antigens and B antigens. And so that's why you're a universal donor as a type O blood type, but you're a universal recipient as a type AB blood type. So now going into RH blood types, it's a little bit different thing, a little bit different ball game. The RH system is another important factor during blood transfusion. In the RH system, spontaneous occurrence of agglutinins almost never happens. Instead, the individual must first be exposed to an RH antigen, usually through transfusion of blood or pregnancy. When RBCs containing RH factor are injected into a person without the factor, anti-RH agglutinins develop and reach a maximum concentration within about 2 to 4 months. On multiple exposures to the RH factor, the RH negative person eventually becomes strongly sensitized to it. The mismatch of the RH factor blood leads to agglutination and hemolysis. That is to say, typically when we talk about AB blood, you're exposed to AB blood unwittingly to such an extent that you will develop agglutinins against it, a reaction towards it when you're first quote-unquote exposed to it. Because really you were exposed to it long before, and that's why you have a negative reaction to it when you actually take a transfusion of, let's say, A blood and you're type B. But with RH factor, typically it's not common enough for you to actually be previously exposed to it unless you were, for example, a pregnant mother and your child had RH factor or something like that. Typically you would not be exposed to it, and so you wouldn't necessarily have a strong reaction to it initially because you haven't been exposed to it before, but you would eventually be sensitized to it if exposed enough. Now, erythroblastosis fetalis, a disease of fetuses and newborn infants, is characterized by progressive agglutination and subsequent phagocytosis of RBCs. In a typical case, the mother is Rh negative and the father is Rh positive. If the baby has inherited the Rh positive antigen from the father and the mother has developed anti-Rh agglutinins in response to this antigen, these agglutinins can diffuse through the placenta into the fetal circulation and cause RBC agglutination. Not good. Transplantation of tissues and organs. An autograft is a transplantation of tissues or whole organs from one part of the body to another. An isograft is a transplantation of an organ from one identical twin to another. An allograft is the transplantation of an organ from one human being to another. A xenograft is a transplantation of an organ from one species to another. So again, auto from yourself, iso from a twin, allograft from the same species, right? And a xenograft xeno implying alien, foreign, something outside the box, kind of something different. It's a different species, basically. In the case of autografts and isografts, all cells in the transplanted organ contain virtually the same antigens and survive indefinitely if provided with an adequate blood supply. In the case of allografts and xenografts, immune reactions almost always occur. These reactions cause the cells in the graft to die within 1 to 5 weeks after transplantation unless specific therapy is given to prevent the immune reaction. When the tissues are properly typed and are similar between donor and recipient for their cellular antigens, successful long-term allograft survival can occur. Simultaneous drug therapy is needed to minimize the immune reactions. Tissue typing is performed to identify the human leukocyte antigen complex of antigens. The most important antigens in human graft rejection constitute a complex called the human leukocyte antigen, HLA, antigens. Only 6 of these antigens are ever present on the cell surface of any one person, but more than 150 types of HLA antigens exist. This number represents more than 1 trillion possible combinations. As a consequence, it is virtually impossible for 2 individuals, with the exception of identical twins, to have the same 6 HLA antigens. And that's important too, because when they're talking about isograft, they're saying, oh, a twin. They mean an identical twin. Because if they're a fraternal twin, then they're just the same thing as like a sibling, basically, a standard sibling. It's only identical twins where they are genetically almost identical, right? They're almost genetically the same. And so they will have the same HLA antigens. HLA antigens are present on both white blood cells and on the cells of tissues. Some of the HLA antigens are not severely antigenic. Therefore, a precise match of antigens between donors and recipients is not essential for allograft survival. But the best results occur in persons with the closest possible match between donor and recipient. Prevention of graft rejection can be accomplished by suppressing the immune system with 1. Glucocorticoid hormones, which inhibit genes that code for several cytokines, especially interleukin 2, an essential factor that induces T cell proliferation and antibody formation. 2. Various drugs, such as azathioprine, that are toxic to the lymphoid system and therefore block formation of antibodies in T cells. 3. Cyclosporin and tacrolimus, which inhibit the formation of T helper cells. These drugs are especially efficacious in blocking the T cell-mediated rejection reaction. Or 4. Immunosuppressive antibody therapy, including specific anti-lymphocyte or interleukin-2 receptor antibodies. And that is the end of Chapter 36 of Guyton and Hall's Pocket Physiology. It's a relatively short chapter, but if you were listening in, thank you for listening.